All right, ladies and gentlemen, welcome. First of all, this is like there won't be a survivor's photo, but thank you for making it all the way to the end. Listen, once again, my name is Giles, and I am very, very excited to chair this final session, and really it's the second keynote from Karen Mohlke. And she is one of the, I hope you forgive me for using the word doyen, but one of the doyens of diabetes human genetics. Did a PhD at the University of Michigan, a postdoc at NIH, and then has been at the University of North Carolina since then, where amongst the things she has done was to be one of the initial pioneers way back when, of using these exome arrays for diabetes genetics. But it is a great pleasure to actually introduce Karen. Thank you so much for agreeing to come across the old pond to speak to us and also be our closing keynote. Thank you so much.
All right. Thank you very much. I really appreciate the organizers giving me the opportunity to come and present our work. So I'm going to talk today about genetics, human genetics, linking human disease to the mechanisms that are underlying the associations that are identified. So we all know, we've heard about throughout this meeting, the various tissues and processes that play a role in metabolic dysregulation, these tissues as well as others. And we know a lot of the players, a lot of the proteins, a lot of the genes that are involved in these various processes. But there's a lot that we still don't know. So we use a genetic approach to try and understand what those players are. So I'm going to focus today largely on adipose tissue and on liver tissue. So by a genetic approach, which really introduced a lot earlier this morning, we're looking at genetic variants, such as the T to C variant here, and their association with metabolic traits. So this has been a well-established analysis now for some 15 years. So for example, if we look at genetic variants associated with plasma alanine aminotransferase levels, we can observe a genome-wide association study analysis where the variants that are found on these plots are showing evidence of association, genetic variants in a region that are associated with variation in these alanine aminotransferase levels. However, as we've also heard, a lot of these, if I zoom in here and look at now only 150 kilobases instead of the 3,000 megabases, each dot still a genetic variant. I'm going to show a number of plots that look like this. When the strongest variants are not located in protein coding regions, then the question is, what is the underlying gene? What are the variants that are driving the association? And can we develop some testable hypotheses so we can validate those links and really understand the basis of human disease? So the big questions are, what are the genes that are responsible for the association, and which variants affect gene function? So the strategies that we use to try and dissect this association between the genetic variants and metabolic traits are first to focus on gene expression levels. So in the same way that we ask whether genetic variants are associated with a trait in humans, we can ask whether they're associated with the level of gene expression of a given gene in a given tissue. And so shown here, the T and C alleles, one of them is associated with higher expression, the other with lower expression of a given gene. And then to understand how those associations may come about, we also look at chromatin context. So chromatin context varies between cells, cell types, tissues. It also varies between individuals, as I'll show in the later part of the talk. And most of the time when I'm talking about chromatin context, I'm going to be talking about the assay for transposase-accessible chromatin. So shown here, the DNA normally is wound up tightly around the green histone proteins, but in some places it's unwound and accessible to the blue transcription factors. And so we identify what those regions are, isolate those sequences, map them back to the genome, and they show up as peaks. Okay, so we have GWAS that are the variants associated with traits, and we're going to integrate that with expression quantitative trait loci, that's the association with gene expression, and the chromatin accessibility QTLs. One more bit of terminology before I get too far. Is this use of my use of the words for loci, signals, meaning association signals, and variants. So here in this example plot that's shown, we can look at the strongest associated variant in the region, call that a lead variant. The other variants that are located right near that are shown in colors, are found in often the same pattern across individuals. That's shown by this linkage to disequilibrium coloring. That represents one signal. So in the old days, we used to say, oh, take the lead variant, map out a region around that, call that a locus, and assume that it's all acting in the same way. But here we have another sort of set of variants there. They're in dark blue. They're not correlated with the first set. That's a lead variant for another association signal. So in this case, this is type 2 diabetes GWAS data from East Asian population, and the two signals may be acting on two different genes underlying them. Okay, so I'm going to tell two stories today, and the first one is going to focus on adipose tissue and expression quantitative trait locus analysis. So this is a collaboration with investigators at the University of Michigan and King's College London, including Corinne Small, who's in the room today. And we performed a subcutaneous adipose EQTL meta-analysis combining together 5 data sets to have more power to identify the genetic variants that are associated with gene expression levels. So here are the 5 studies that we combined together, 2 from the MetSim study, 1 from Fusion, 1 from Twins UK, and 1 from GTEx. When we started doing this analysis, one thing we first identified, that figure did not transfer well, is that there are differences in the quality of the samples across the different studies. So you can't see the coloring to know which study is which, but this is AdipoQ expression levels, which are a proxy for adipocytes present in the tissue. So those levels are higher in women than in men, and the cleaner studies, so Twins UK, it's furthest on the right. It's the cleanest data, punch biopsies, lots of adipocyte content. GTEx, the worst. Over on the left, in fact, there are some samples that had such low AdipoQ that we discarded those samples and had stronger results. So we discarded about 15% of the samples from GTEx and had cleaner EQTL data for looking at adipose analysis. Okay, so each individual study, now they're color-coded with a dot there on the left, and you can see in the plot on the right, in the lower left corner, those are the number of genes in asterisks, the number of signals in dots that were identified in each of the individual studies. And then in the meta-analysis, you can see that there's more than twice as many signals are identified when combining the data together than when looking at them separately. So for this analysis, we specifically went in and to try and identify the separate signals. We used software called Apex, newly identified, new strategy to be able to separate those signals out, and it helped us identify them together. So one thing we did then is look at the characteristics of the signals that are identified. We have so many more association signals in this meta-analysis than previously. What are the characteristics of them? So for time, I'm just going to show you one of those characteristics with respect to signals, and that is we look now at the genes that there are hundreds of genes that have 5 or more association signals for gene expression. So maybe there's a couple promoter variants, some enhancer variants that are all separately influencing the expression level of the gene. Maybe they act in the different cell types. We're looking at bulk tissue here to have their effect. What we see is that on average, the first signal is close to the promoter. The second signal, third signal, they are located further and further on average away from the promoter. So there's a lot of promoter bias in a small EQTL study. You're tending to find those promoter signals first and then later can identify the signals that are located further away. We also looked at some of the characteristics of the genes identified. So here, Dongmeng Wang analyzed the highly expressed genes compared to the lowly expressed genes. And you can see that genes that are more highly expressed, the curve at the top on the left, we identify more and more signals for them. That makes sense. The more highly expressed you can account for, you can see the effect of the first signal, account for it, and look at the effects of the additional signals. In addition, on the right, the genes that are more heritable are more likely to have more signals that we can identify in this analysis. And then we compared the location of the variants for the signals and did an enrichment analysis looking at the roadmap epigenomics chromatin states for adipose tissue. And you can see that especially for first signals, that there's a very, very strong enrichment for promoters, that there's a bigger drop-off towards the secondary signals. There's still enrichment for promoters, absolutely, second, third, and fourth signals, still quite strong, but it drops off dramatically. Enhancers, there's also enrichment on the first through the fourth signals. Okay, and so then, as described previously, what we really care about is the human disease. So let's use these eQTLs to understand the GWAS signals. So if we were to just take our GWAS variants and evaluate them and say, here's my GWAS signal, I have a variant, let me go look up and see whether it's associated with expression level of some gene. I look it up, I find some eQTL, I go over and say, wow, that's really strongly associated. Maybe those are driven by the same signal. However, those patterns are quite different from each other. They're probably not driven by the same signal. So that's where the methods of colocalization that we heard about this morning derive from. And there's various ways to perform these methods. The idea is we're looking at the variants associated with traits, the variants associated with gene expression level, and hypothesizing that maybe the variant acts through the gene expression to affect the trait. So one thing that we realized in this process, we had so many signals, they're located pretty close to each other, is that we needed to statistically separate the signals out to have more accurate methods for colocalization, to better identify whether a particular GWAS signal and a particular eQTL signal are the same. So our eQTLs, we'd already done that. We'd used this APEX and done this sort of effective conditional analysis to identify the distinct signals. For the GWAS data sets, we then went and used the summary statistics from each GWAS data set we analyzed, ran our own GCTA analysis using the summary statistics to separate out the signals. Right? So GWAS these days, there's thousands of signals for some of these metabolic traits, and often they're located quite close to each other. So if we separate them out, condition on the signals that are nearby for any given signal, then perform the colocalization, we get more accurate results. So here's our, we did this for 28 traits. I only put 13 on the slide so that it would be more readable. So here are some of the GWAS traits that we analyzed, and the numbers of colocalized signals. So the GWAS signal and the eQTL signal have a shared pattern of association. So you can see that there are hundreds present for some of these traits for which there are hundreds or thousands of signals identified in GWAS. If we look at these are associations between the variants with a GWAS trait and with a eQTL. Sometimes a GWAS signal is associated with more than one gene. Right? These are regulatory elements that are maybe having an effect on 2 or more nearby genes, so the number of genes can be higher than the number of signals. When we count through and say, well, how many of these did we identify by just looking at the primary signals, the ones that are easier to identify in the first place, and how many come from the secondary signals, you can see that about a third of the signals, the colocalization is identified with a secondary or a later eQTL signal. So doing that separation of the signals, we identified effectively 50% more colocalizations than we would have observed otherwise. Okay, so lots of genes, lots of colocalizations. Which ones do you believe? If a variant is associated with expression level of more than one gene, okay, we're like not being so helpful to the drug companies that are trying to figure out which gene might be responsible. So there's functional follow-up that's needed. So we did a cis-Mendelian randomization method using when there are pairs of signals that are associated with a trait and pairs of signals that are associated with gene expression level, and use those allelic series to look at Mendelian randomization. So the method that we applied here is called MR Locus, developed by my faculty colleague at UNC, Mike Love. And we applied this to the nearly independent conditional EQTL signals. We defined that based on R squared. We're now evaluating whether we also have to be incorporating D prime into our defining things as nearly independent, because we want separate information to know how strong the support is for the evidence of association. So what these plots show is on the left is we're putting the effect size of the EQTL on the x-axis, the effect size of the GWAS association on the y-axis, and then each co-localized signal gets a dot. And this analysis estimates the gene-to-trait slope, and it accounts for errors in both directions. That's what makes it different from other methods of Mendelian randomization. So the plot on the left shows a very nice evidence of mediation, with the signals all showing a pretty clean gene dosage to trait effect. Higher expression on the x corresponds to higher trait value on the y. The middle plot, okay, there's variants there that are showing generally in the same direction, but it's not a very precise measure of the dosage effect. And the third plot, the one on the right, there's no evidence of mediation around. So here's an example of what I'm talking about. There's two plots here, the waist-hip ratio GWAS data on the top. Now the signals are colored so that one signal, the primary signal, is in red, and the secondary signal is in blue. And you can see that the two GWAS signals correspond to or co-localize with the two EQTL signals for the gene ZNRF3 shown below. We can then use those and plot those conditional effect sizes on the plot shown on the right. It's a kind of a wide variation there, but the direction matches what's understood about the protein function. So this is a gene that's been shown previously to negatively regulate Wnt signaling activation. There's a lot of Wnt signaling involved in waist-to-hip ratio and patterning. And so this corresponds and suggests that ZNRF3 may be the gene underlying this particular association. And here's another example. This is the gene PDE3A. Here we have 4 GWAS signals on the top for HDL cholesterol levels, 4 different colors. And there are 4 signals on the bottom for variants associated with gene expression of the PDE3A gene. And all 4 of those signals meet our statistical thresholds for colocalization. We plotted only the independent ones based on R squared on the plot on the right. And you can see that the association, higher gene expression, is corresponding to lower HDL levels. PDE3A has been known to contribute to ventricular remodeling, a role in adipose tissue not necessarily so clear. Maybe this has to do with some of the cell types that are involved in adipose tissue, but a lot remains to be learned about how this could be functioning. Those are 2 new examples. Here are 4 pretty well-established, we'll call these the positive control examples, where the signals that are associated with the LPL gene variation corresponds to coronary artery disease risk quite cleanly. Many signals matches well-established relationship between this gene and trait. The same for GALNT2. The direction of effect is matching what's been shown previously in multiple species about a relationship between that trait and that gene and HDL. Same with EYA2 and RSPO3. So using these approaches then, when we have those allelic series, we have more confidence in naming the genes that we think are underlying the association signals. Okay, so one last piece of integration with those EQTL signals is trying to understand what the regulatory variants might be. So here's an example, a GWAS plot on the left, and only the first signal. So this is the first signal. We conditioned away the second signal, so it's not shown, so it's easier to see the colocalization between the 2 signals. And we investigated the variants that could be responsible for this effect on expression that may affect waist-hip ratio. So shown here is a region of the genome that's mostly SEMA3C across the top here. And the variants that show evidence of association with gene expression in the trait are located right here. We're looking at these compared to some chromatin states for several different cell types and with some attack-seq data for adipocytes and adipose tissue. If I zoom in, you can see with some of these variants, including this one right here in the right of that, within that circle, that the variants are located within a peak that is more accessible in mature adipocytes than it is in pre-adipocytes. So we thought that looked like a pretty good candidate for playing a role in affecting gene expression in adipose tissue. And so, I guess I'll say first that 70% of the EQTL signals that we identified had one or more variant located in a region like this, a candidate for potentially playing a role in gene expression. So then we wanted to validate, does this variant really have an effect on transcriptional activity? And so performed a luciferase assay to analyze that, where we cloned the region that the variant is in with either the G allele or the C allele, and performed luciferase reporter assays in a human white adipose tissue-derived cell line. And you can see there's really clean. It's a very strong enhancer compared to empty vector. This is our fold change. And there's a very clean allelic difference in both the forward and reverse orientations with respect to the promoter, strongly suggesting that this variant can affect transcriptional activity and so could be responsible for the effect on gene expression and then maybe also on the effect on the trait. So we can use data like these to build the mechanisms to understand how the DNA variants are having their effect on genes and leading towards disease. So for this section, I've shown you that EQTL, our meta-analysis, identified more than twice as many signals and almost twice as many genes than were detected by the 5 studies individually. Most EQTL data sets are underpowered, is what we're learning. We have not saturated at all the number of signals that can be identified, and larger studies will continue to identify more signals so we learn a lot more about how gene expression can modulate, how variants can modulate gene expression. Those non-primary EQTL signals, the ones identified later, showed less promoter bias. And when we compared the EQTLs to the GWAS data, we found more than 1,800 candidate genes for cardiometabolic traits. Including those conditionally distinct signals helped us identify another 50% more genes. So we value this approach to further dissect all of those genetic signals that are influencing the trait. And then when we use the allelic series, the pairs of variants or the triples of variants that are showing independent links of variants to genes, then we can estimate the dosage effect, how much that gene is having an effect on that trait. Okay, now I'm going to switch gears and turn to, that was adipose tissue and focused on gene expression levels. Now I'm going to switch gears and turn to the liver and focus on the genetic variants that are associated with chromatin accessibility. So now we're going one level maybe deeper, one level closer to the effect of the genetic variant to understand how it can influence gene expression. So you're probably aware that chromatin context can differ by cell type. We're showing where chromatin context and the accessible chromatin varies by individual and their genetic variants. So I'm going to kind of mix together 2 studies as I talk about this. One is published where we looked at 20 individual liver samples from 20 individuals, and we assayed chromatin accessibility, identified variants, and did many of the same kinds of analyses that I'm going to show. But I'm going to mostly pepper in the unpublished data that comes from liver tissue samples from 138 individuals where we did this and just have a lot more power to identify the genetic variants that influence chromatin accessibility. And this work was largely driven by Kevin Curran. Okay, so we're going to map chromatin accessibility, QTL loci, and we did this in 2 separate analyses. One, we focus on only looking 1 kilobase away from the summit of an attack-seq peak. So we're talking about the regulatory element, and we're looking for genetic variants that are basically in or very close to that region. However, we also want to be able to do colocalization analysis, and that requires having a more broad set of genetic variants to better understand what the signal looks like. So we also did a 1 megabase analysis, looking at all the variants within a megabase of a given peak and learning about the evidence of association. So when I say we're doing a chromatin accessibility QTL, we're basically asking whether there's a variant at which one allele, the homozygotes for one allele, there's not much accessibility. The homozygotes for the other allele, there's a lot more accessibility. And the heterozygotes are in the middle. So when we do this, we had around 350,000 regions of chromatin accessibility across the liver tissue samples in these individuals. And about 35,000 of them with a FDR of less than 5% showed evidence of a variant that's associated with the amount of accessibility. To give you an example of what this means, here are the tracks, or the genome, sort of the profiles for 9 different individuals across a given region. So you can see there are many regions of the in this area of the genome that look very similar across all of the different individuals. However, there are some regions that differ, like this one right here. And shown on the left is the genotypes of a variant that's located right in the middle of that peak. So you can see the individuals with the TT that are homozygous for the T allele have stronger evidence of accessibility than the individuals with the CC genotype. It's hard to even tell if there's accessible region there. So one way that you can have differences in chromatin accessibility due to genotype is that a transcription factor may bind preferentially to one allele than the other. So we looked for which transcription factor motifs are preferentially disrupted by the CAQTL variants. So shown here are the transcription factors that showed the greatest difference in their CAQTL status compared to peaks that are not CAQTL. You can see HNF1 alpha, not surprising, it binds all over the place, like 100,000 regions across the liver genome, is found quite a lot within our CAQTL associations. There's also some pioneer transcription factors in here that are maybe playing a role in helping the variants become, the variant regions to become accessible. So these are suggesting the transcription factors that may play the biggest role in their effects on chromatin accessibility in the liver, and then subsequently effects on gene expression. Okay, so we find these peaks, they're genetically driven, but we'd really like to know those peak to gene links. Which genes are those peaks acting on? So before I show you the 4 methods that we use to try and do this, I want to talk about one of them. And this is looking at coordinated attack-seq peaks. So here is a region of the genome, and you can see that in the left-hand side of these 3 plots, that there are some very similar similar chromatin accessibility regions, regardless of the genotype that I haven't shown you yet. So these parts of the plots look very similar across the 3. But here in this purple, you can see that there's a peak that is much more strongly accessible in the homozygotes for this, the alternate allele compared to the homozygotes for the reference allele. This is the variant that has been associated with LDL cholesterol levels, has been really implicated through many different analyses at the variant, the gene, and the organism level to be playing a role in LDL cholesterol through its action on the SORT1 gene. So if you look at all of the peaks in this region, you can see the same sort of coordinated effect on the association. So all of the peaks, not just the one here, but all of the peaks represented here are stronger, more accessible in this homozygotes here than they are in the other allele. So this peak, stronger, gets weaker, same over here and over here. So these include 2 promoter peaks at SORT1. So when we observe more accessible chromatin at a variant-containing element, say the GWAS variant, and we see a coordinated effect with promoter chromatin accessibility, then we use that as one way to potentially be linking those peaks to their effects on given genes. Okay, we used 4 methods to try to link those peaks to their genes. I just showed you number 3 here, the promoter coordinated peak. Here, in each case, the chromatin accessibility, the variant located in the peak is the purple one. Here we see this pattern with promoter peaks. We also use nearest gene. If it's in the promoter, then it's maybe more likely to be affecting that gene. We use Hi-C data from liver and from hepatocytes. It's very promiscuous. There's a lot of links that show up there. And then finally, we use CAQTL colocalization with EQTL. So the EQTL gives the gene. So if the signals are looking the same, then we say maybe the CAQTL variants are acting through the effect on that gene. So here are the number of peak to gene links identified by each one of these different methods. They all have their pros and cons. Some of them are very limited to the locations very close to the peak. Some of them are limited to the locations further away from the peaks, like the chromatin, the Hi-C loops, where you don't look at the signals that are so close to the peak. Together, they identify 13,000 of the peaks we were able to link to at least one gene. So there's a lot of work still to be done in the peak to gene linking arena, and to figure out which of these methods are the most valid. Okay, so we have our peaks. We've linked some of them to genes. We, again, really care about human disease. So now we're going to co-localize the CAQTL signals together with GWAS signals. So here are the GWAS traits that we selected for this analysis. We focused on traits more likely to have their effects through the liver, and analyzed the colocalization to be as rigorous as we could in terms of trying to identify which signals could be responsible or could be shared between the two types of data. So here we show the number of GWAS signals that were close enough to a chromatin accessible region to test for colocalization, and then the number in the middle that showed colocalization between the chromatin accessibility and the GWAS trait, and then the percentage of those signals that are colocalized. So as you'd expect, the traits that are more likely liver-oriented showed a higher percentage of those signals that show colocalization. So all of these now offer candidates, and now we're at the candidate variant level, not just at that sort of the gene level, but now we have candidate variants that we can potentially evaluate for their effect. So here's, as an example, for alanine aminotransferase, we have 44 signals that were co-localized between the CAQTL analysis and the GWAS analysis. So in terms of trying to validate some of these associations, I'm going to just walk through one example. So now there's 3 plots shown here. The upper left is the GWAS data for liver enzyme levels. This is in more than 100,000 people. The bottom is liver EQTL data for the EFHD1 gene. This is in around 1,000 people. And then on the right, the bigger plot is the chromatin accessibility QTL data. And this is actually in the 20 people. So the same evidence of association is observed. When you only have 20 people, you get a little bit less resolution to the evidence of association. The lead variant for the chromatin accessibility QTL is amongst the strongest variants for the GWAS data and for the EQTL data. And this peak region, so now I'm showing what this region looks like, what this CAQTL is. The individuals here that are homozygous for the T allele are the more accessible. So here's the regulatory element. It's located in an intron of the EFHD1 gene and links to the promoter through promoter capture Hi-C data. So we have two ways of linking this peak to the gene, the eQTL and the chromatin capture data. So then we asked, is the variant that's underlying that CAQTL, could that variant change transcriptional activity? So same as before, doing a luciferase reporter assay, cloning the CA peak with the two different alleles in front of a minimal promoter and luciferase. We observed that indeed this is a strong enhancer element and that there's substantial allelic differences showing that just the change of that one nucleotide can change how much transcriptional activity we observe. This is now in HepG2 cells, in, again, both orientations, providing support for this variant. So now we have a variant and a gene, a direction of effect together that could be influencing the trait. Like, well, how does that variant have its effect? So we notice in this region that of the chromatin accessible peak that there's ChIP-seq data from ENCODE from HepG2 cells for quite a number of transcription factors, including FOXA2, where the motif is predicted to be disrupted by this particular variant. So we ran an electrophoretic mobility shift assay to identify if there were any proteins that bound differently to the sequence, the DNA sequence, right around that variant, around 17 nucleotides. So you can see in lane 6 that there's a band that's present, not in lane 1. That corresponds to a protein that's binding to this short probe when the T allele is present and not when the A allele is present. And further, when we add an antibody to FOXA2, so we have got the DNA and now the protein and now an antibody to FOXA2, we observe a super shift. The variant, the band becomes weaker here and stronger and larger up here, providing even stronger support that FOXA2 could be the transcription factor that is influencing this effect on gene expression. Okay, and then one more way to try and validate the link between the peak, because we're identifying peaks with the CAQTL, to the gene, and that is to use CRISPR-I. This is a lot like what Kelly just described to us. So here we have this element that's located in the intron. We created a cell line that was stably expressing an inducible, a dCas9 with a Crab domain on it, so that when we introduce guide RNAs that are targeted to the enhancer element or this regulatory element, we're bringing repressive domain action to that region. And so when we go measure then the gene expression level after using these guide RNAs, if we see the gene expression level change, then we say perhaps that element is having its action on that particular gene. So here we cloned 5 different guide RNAs together as a pool with lentiviruses, introduced those into the cells, and then used quantitative PCR to measure the expression level of both the target gene, this is the one that we're predicting, EFHD1, as well as 2 other nearby genes to see if there's an effect there. So here you can see that there's a modest effect, a modest reduction in the expression level of EFHD1 when we use a pool of guide RNAs to the enhancer compared to a pool of control non-targeting guide RNAs. So that modest effect is perhaps reflects, this is one enhancer that has its effect on gene expression levels. You would imagine the promoter has an effect on gene expression levels and the other enhancers as well. So 20% might be our expectation. It does mean we need to be doing a lot of replicates to be able to have confidence in some of these kinds of results. When we compare to the 2 other genes nearby, we don't see an effect, a significant difference for this gene over here. Gosh, there's an awful lot of variability going on with the gene over here. We don't see a significant effect there either. So we can take together all of these different types of data and propose then a mechanism for how this GWAS signal could be having its effect. How do the DNA variants lead to an effect on that trait? So we've shown that with this particular variant and allele, that the T allele here corresponds to increased chromatin accessibility. That's the CAQTL data. It corresponds to increased FOXA2 binding. That was predicted from the motif and then validated with the EMSA result. Corresponds to increased transcriptional activity. That was hypothesized first from the EQTL data and then validated with the luciferase reporter assay. It corresponds to higher EFHD1 expression in the liver. That is the EQTL data. And then this is all associated, the same sort of direction of effect with increased levels of alanine aminotransferase from the GWAS data. Obviously, a lot of steps in between the how does the variant affect the gene to how does the gene get to the trait. That's a lot more work to be done, but this is providing strategies to help dissect the GWAS signals to get to the traits. So in conclusion for this section, DNA variants can have very strong effects on chromatin accessibility. We are now as close as you can get to the effect of the DNA variant. The effect on chromatin accessibility, very close. The effect on gene expression, next most close. The effect on protein levels, next most close, perhaps. The effect on phenotypes, getting further away. So even in only 20 individuals, we are able to identify thousands of C8QTL. It takes more and more samples the further away you get to be able to identify the genetic effects on different types of molecular traits. So here with 138 individuals, we found 35,000 regions across the genome where the variant is influencing the regulatory element accessibility. C8QTL corresponds to sample size. It also corresponds to read depth. When we sort of downsample our read depth, we find less and less evidence for the chromatin accessibility. The larger the sample size, the more that we would be able to identify. We're able to predict target candidate genes for around 38% of the peaks. This is pretty poor still. There's a lot still to be done in trying to understand how to link the elements, the regulatory elements to the genes. However, the C8QTL are strong candidate regulatory variants for GWAS and EQTL signals. So integrating all of the data together, the C8QTL, the transcription factor motifs, the target genes, that lets us build these predicted functional mechanisms, have evidence of direction of effect across multiple of the modalities to have more confidence in how the mechanisms may play out. And then finally, single cell analysis that we have ongoing are going to help also elucidate cell type. So with liver, about 60% of the cells are the hepatocytes, so the effects that are happening in hepatocytes we're probably finding just fine in the bulk tissue analysis, but the ones that are affecting the LSECs, the other cell types, the less common cell types, could be completely masked in a bulk tissue analysis. Okay, so overall, using these genetic approaches, we identify hundreds of loci that are associated with metabolic traits. We can then use gene expression in lots of different of the relevant tissues to identify some target genes, their potential directions of effect to better understand what those targets might be, and then use the chromatin context to be able to identify potentially specific variants, either because their variant is located in the region of chromatin accessibility, as I showed with the first story, or that there's a genetic association with accessibility, as I showed in the second story. And together, then, using these mechanisms, hopefully we end up being able to lead to reduced effect, better targets, learn more biology to be able to reduce the impact of metabolic disease. All right, I'd like to thank the many individuals that were involved in both of these two projects. The first one with the gene expression levels was a collaboration with Corinne and a group from Twins UK, from Fusion investigators, and from our MetSim investigators. And then the liver tissue samples were from healthy but deceased donors. They were selected for having relatively little drug use prior to the sample collection. Those were obtained from St. Jude. And appreciate all the consortium data that went into being able to do an analysis like this. Great. Thanks very much.
Thank you so much for that, Karen. That was fabulous. I had a little bit of PTSD because I spent a decade of my life working on the FTO locus. And the less said about that, the better. So anyway, but it's just fantastic to actually see the sophistication of which we've actually now arrived at, to try and get these statistical signals after all and link them to actually to biology, to human biology, and that is fabulous. Right, folks, we have time for questions. Anybody the question? Why don't we start with Anna over here?
Thank you, Karen. That was fantastic. Now, Giles, hold on. I'm going to take you back to FTO.
Oh God!
Sorry. So, the number of EQTLs that you found in the adipocyte study is fantastic, really exciting. But thinking about FTO and the role of the developing adipocyte, what are your thoughts there on what we're missing by working with, firstly, bulk tissue and tissue from the developed, rather than looking at it during differentiation or at a single cell level?
Right. I mean, there are several different directions there that you point out. So bulk tissue from, these are all, by the way, except for GTEx, people who walked in to the clinic, donated a tissue sample, and then walked out, so on that spectrum. So there will be different genetic effects amongst diseased individuals. Now, some of those are probably just masking what the real genetic effect is, like the direct effect of the variant on the gene expression. But some might be providing the context, like these are the inflammatory, there's more inflammation happening. And so then those transcription factors are turned on. You have a better chance to be able to detect that. So it could be that diseased individuals will show more. If we could obtain samples, maybe through iPS cells derived, and be able to look at the different times of differentiation, and then capture and perform the EQTL study at those different states of differentiation, I bet we will learn more. I think there's a bunch that we're missing there. When we look at those chromatin regions from the pre-adipocytes compared to the adipocytes, right, some don't change very much, but some change quite a bit. And I think figuring out which ones are having their effect on differentiation, that would be a great way to hone in on those, I suppose. The other is just other perturbations. So let's look at the genetic effects on gene expression, but now perturbed with insulin levels, palmitate levels. We could do that in a dish that maybe we can't control quite so well in the human individuals. Harder to maybe get the large numbers of samples to have the most power. And then absolutely at the single cell level, I think we'll be able to learn with large enough data sets. We'll be able to learn not only about what's happening in each cell type, and again, like with adipose tissue, most of the cells are adipocytes, but can we learn what's happening with the mesenchymal stem cells? Can we learn what's happening with the endothelial cells and what those genetic effects on gene expression are? That'll absolutely help. And then even within a common cell type, the different cell states that are happening, if we can take the cell type adipocyte and figure out what the different groupings of cells are, then we might be able to determine EQTLs separately amongst those. So plenty of work to be done.
Yeah, fantastic answer, very thorough. So just to follow up, if I may,
please.
What is in the GTEx then? If it's not adipocytes, what on earth did they take?
Yeah, so the GTEx subcutaneous adipose was taken from the leg. There's a lot of just fibroblasts there. You can go look at the images online. That's what gave us the confidence to just throw away the samples that had the low adipo-Q expression levels. I mean, GTEx is great, right? And there's lots of tissues. And how do you get lots of tissues? You get them from, by being able to process samples quickly from lots of different places and all. So, maybe that's where, when you care about one tissue and you focus on one tissue, maybe get a little more reproducibility in the data, the quality of the data than, across.
Question over there?
Hi, very nice talk. I have two questions. The first one is regarding the first part, where when you're showing the secondary or conditional signals are more enriched in enhancers, probably that is expected because you're moving away from the gene. So I'm wondering if you have compared tissue sharing, whether the promoter signals are more shared across signals and the distal ones are not that much shared and they're very specific to tissue.
Yeah, so we tried to compare the signals that we observe across tissues. So we tried this using EQTL-Gen because it's so large, it's 30,000 individuals, blood. However, they didn't separate out the signals, so it was harder to detect that. We observed many signals that are many eQTL signals that are only detected in the adipose tissue, but it's hard to know whether they're just masked and sort of missing at the moment from the blood data because the signals hadn't been separated out. So I would expect that there's going to be less sharing once we get into the enhancers, because there's a lot of variants in promoters that are shared across cell types that are going to show the same eQTL effect regardless of whether the gene actually matters for that cell type. It's just like if the body has no reason to say, I don't need genetic variation influencing me, then it may still persist. So as we get into the enhancers, and especially because there's more enrichment of cell type specific enhancers with disease, when the disease relevant regulatory elements, I think that there will be less sharing as we get further out.
Sure. So my second question is, when you're trying to link the CAQTLs to the genes, like target genes of the CAQTLs, in a way that eQTLs are chromatin QTLs, or they have to affect the regulatory element to show effect on the gene expression changes, right? So when you have a CAQTL that doesn't have an eQTL, do you think those CAQTLs are acting in a context that we haven't captured in eQTLs?
So that's the most obvious explanation. So we are really interested in those. What are the CAQTL that don't have their effect on gene expression? I will say the eQTL data we're using for liver, only about 1,000 individuals, and was actually derived from microarrays, not even RNA sequencing. So it's probably super underpowered. So that's my first conclusion, is that the CAQTL that are not eQTL, we're not observing the eQTL. But my second conclusion is, yes, the context of the samples that are used for the eQTL analysis.
Thanks.
Second time you've hit me with PTSD was the mention of the word microarray. Anyway, so I think we have a question online. Question online first? Question online first?
Oh yeah, I've got a mic, haven't I? Thanks. Thanks for reminding me. So this comes from Ivan Karkemo, Oriv, and he asks, can you comment on how current in vitro adipocyte or hepatocyte cell lines actually reproduce the biology of primary cells, and how that conditions in vitro functional validation for the candidate causal genes? So how does it compare with, when you're taking biopsies compared with the cell line-based assays?
Right. I mean, it's a cell line. These are models. The biology, I think, is the hardest part when expecting some, so when we use a cancerous HepG2, cancer, hepatocellular carcinoma cell line, to look at the effects of a variant on gene expression, all we're really asking is that the transcription factors that matter for that effect on gene expression are present in the cells. Maybe not too bad. Once we get into the biology effect and saying, when we knock down that enhancer and then ask what's the effect on biology, now we're into much more challenging territory. Yes, using a cell line that has the best biology is going to matter, most, because what we really care about is the biology. I'm getting us from variant to effects on gene expression, but there's so many more steps after that.
Thanks.
Claudia, you got a question?
Oh, I think maybe I have a micro as well. Yeah. Sorry. Absolutely amazing. Thank you so much. So it's a very simple question, but I mean, given that you've done all the hard bits, the simplest thing is in terms of expanding the diseases that you consider. You looked at the traits, presumably many of those, what you say, liver-related. It's a metabolic disease meeting that were powered because they're continuous traits. But actually, the ICD chapter codes for liver being so central is huge. So that's an easy thing to do and to kind of see the relevance across so many other disorders. Are you doing this? Are you planning this? Or
So I think you're asking whether we're using data sets for the disease traits, not just the continuous
so
traits.
Expanding it to other liver diseases, I guess is what I'm saying.
Yes, so yes, I mean the data sets for like NAFLD, if you really want to define NAFLD properly, there's just many fewer loci, most of which are already captured when you're using those continuous traits. They're present. We did also go look at the sort of drug metabolism genes to try and understand what are the genetic effects of variants on those drug metabolism, just as a separate kind of strategy to understand how biology might work. I think that the potential to learn about any other kind of gene. You don't have to care about GWAS, you just have to care about liver, for example, to be able to make use of what are the regulatory elements that matter.
Fantastic. Okay, last question, because we're in the red minus and people want their fish and chips. Vegan options are available.
Great talk. So some therapeutics are targeting transcription factors to activate them. For example, TZDs target PPR gamma to activate them in the adipose tissue. Some of the more promising drugs for liver disease are FXR agonists. Could you predict, based on your chromatin accessibility data, which genotypes will respond better for transcription factor activating drugs compared to, general population?
I think you'd have to get a more comprehensive setup. Like, we're identifying a lot of the time one regulatory element in a region, maybe two regulatory elements in a region. Perhaps when we have the more expanded data, like with the EQTL, we found many, many more signals of association that affect a given gene with a larger sample size. I think you'd want that more comprehensive data to be able to be getting into the making good predictions based on those variants. Maybe more comprehensive. Maybe with these just beginning evidence of association, you could do some of that prediction, but I think it's maybe a little bit off.
Thanks.
All right, and with that, can we thank you so much, Karen. That was fabulous.